Morning students, today we are going to look into the types of biosensor part 2, and we know the course outcome for the fourth unit is that students will be able to acquire the basic knowledge on biosensors, organic chemistry and spectroscopy. As a learning outcome for this lecture is, at the end of this lecture, students will be able to acquire basic knowledge on types of biosensors. We already listed that the biosensors could be classified based on bioreceptors and transducers. Based on the bioreceptors, we have enzyme biosensor, microbial biosensor, affinity biosensor, and antibody biosensor. And based on transducers, we have optical, calorimetric, gravimetric. Actually, this is colorimetric, calorimetric, pyroelectric, electrochemical, where we include angulometric and potentiometric and conductometric also. In last class, we looked into calorimetric, optical biosensors, and piezoelectric biosensors. So today, we look into the electrochemical biosensors, resonance biosensors, and ion-sensitive biosensors. In the case of electrochemical biosensors, the measurement is based on the electrical properties of the solution, and this is used for hybridized DNA binding drugs, and glucose concentration. Whereas conductometric sensors is based on electrical conductance or resistance of the solution. In the case of amperometric biosensors, this is based on the current measurement. Potentiometric sensors work based on the ramp voltage which is applied, and that will produce a current flow. And this is based on redox reaction. We also have something known as resonant biosensors, where an acoustic wave transducer is coupled with bioelement. So this measures the change in resonant frequency. Ion-sensitive biosensors also we have, where semiconductors FETs and ion-sensitive surface is used, and the surface electrical potential changes when the ions and semiconductors interact. This measures the change in potential, which helps in pH detection. First of all, let us look into conductometric biosensors. As I told you, this works based on the change in ionic species, because many of the reactions in biological systems, they involve change in ionic species. So the ionic species alter the electrical conductivity, we know. And here we are measuring, actually this is known as urea biosensor, and this utilizes immobilized urease. This breaks down urea with the help of urease. So, and this biosensor could be used mainly in dialysis and renal surgery. And the next one is amperometric biosensors. This works based on the movement of electrons. Actually, a constant voltage will be passed between two electrodes, and the substrate or product can transfer an electron with the electrode surface to be oxidized or reduced. And alternate current flow that can be measured here. Best example is clock oxygen electrode. The other one is determination of glucose by glucose oxidase. Here we have first generation, second generation, and third generation. In the case of first generation, we have some practical difficulties. So we go into the second generation, where a mediator like ferrocene is used, but they are yet to become popular. Whereas in the case of third generation, direct removal of electrons from the reduced enzymes, and the electrodes here are coated with electrically conducting organic solids or salts. Blood glucose biosensor is the best example for amperometric biosensors. We all know, like most percentage of the patients are diabetics. So this is like a watch pen. We can use only for single use. and this is coated with silver or silver chloride, reference electrode, and carbon working electrode with glucose oxidase and a derivative of ferrocene as a mediator. And the electrodes are covered with hydrophilic mesh gauze. The disposable test strips are sealed in an aluminum foil and we have shelf life around 6 months. We also have another one that is for accessing the freshness of fish. The accumulation of inosine and hypoxanthin in relation to the other nucleotides indicates how far the fish is fresh, whether it is dead, how long it has been dead and stored. A biosensor utilizing an immobilized nucleoside, phosphorylase, and xanthin oxidase or an electrode has been developed for this purpose. Next comes potentiometric biosensor. Here, change in ionic concentrations are detected by ion selective electrodes as we do in pH electrode. Here, the enzymatic reactions involve the release or absorption of hydrogen ions. We can also have ammonia selective and carbon dioxide selective electrodes. But the potential difference obtained between the potentiometric electrode and the reference electrode can be measured. This is proportional to the concentration of the substrate. The major limitation is that the sensitivity of enzyme to ionic concentrations such as H plus ions and ammonium ions are very bad. So that has to be monitored. We also have ion selective field electric effect transistors, ISFET, are the low cost devices for miniaturization of potentiometric biosensors to monitor intramyocardial pH during open heart surgery. The next type is whole cell biosensors. This is used for multi-step or cofactor requiring reactions. It employs live or dead microbial cells. The advantage is that this is cheaper with longer half-lives and less sensitive to variations in pH and temperature compared to isolated enzymes. But the limitation is that it takes longer period for catalysis and the specificity and sensitivity of the whole cell biosensors may be lower compared to that of enzymes. Selected list of organisms along with the analyte and the types of biosensors are listed in this table. We have next one immunobiosensors. Here, immunobiosensors or immunochemical biosensors work on the principle of immunological specificity coupled with measurement based on amperometric or potentiometric biosensors. You could see four types here, A, B, C, and D. In the case of A, an immobilized antibody to which the antigen can be immobilized antibody to which an antigen can be directly bound. And the second type, an immobilized antigen that binds to an antibody, which in turn can bind to a free second antibody. In the case of third type, an antibody bound to an immobilized antigen, which can be partially released by competing with free antigen. Okay. In the case of fourth one, an immobilized antibody binding free antigen and enzyme labeled antigen in competition. Okay, so we have four types of immunobiosensors. 1 to 3 could be measured by piezoelectric devices. The immunobiosensors using enzymes 4 are the most commonly used. These biosensors imply thermometric or amperometric devices. The activity of the enzyme bound to immunobiosensors is dependent on the relative concentration of the labeled and unlabeled antigens. The concentration of the unlabeled antigen can be determined by assaying the enzyme activity. Moving on to the applications of biosensors, right? We can detect the freshness of food in food industries. We can study the biomolecules and their interaction, drug development, industrial process control, transplantation of organs, clinical and laboratorial diagnosis of diseases, crime detection, detection system for biological warfare agents. And it is also used for glucose monitoring in diabetics patient. And we can also monitor methanol, acetonitrile, phenolics in water bodies. Potentiometric biosensors are intended primarily for monitoring levels of carbon dioxide, ammonia, and other gases dissolved in blood. We can also detect pesticides and river water contaminants. Here they are comparing different electrochemical biosensing like conductometric, amperometric, and potentiometric, how they are working based on the parameter which we measure, applied voltage, sensitivity, etc. And they're comparing the transducers, the advantages and the disadvantages. This depicts the advantages of nanotechnological approaches to biosensor development. It increases in sensitivity of the sensor, enlargement of the operating range of the sensor by extension of lower and upper detection limit, fabrication of small non-invasive sensors that were considered to be impossible in previous technology, then fabrication of sensors by bottom-up approach to simplify process and minimize cost. These are some more applications of biosensors, both clinically and non-clinically. We can also use in food analysis, study of biomolecules and their interaction, drug development, crime detection, medical diagnosis, environmental field monitoring, quality control, industrial process control, detection system for biological warfare agents, the manufacture of pharmaceuticals and replacement organs. This lists the biosensor for agricultural and food industry, where we can detect the viral, fungal, bacterial disease of plants. In food industry, detection of total microbes and food quantification in soft drinks to determine the freshness of other fish, beef, and other food items. Actually, it makes the bacteria glow by an optical biosensor like this. This is a biosensor used for detection of pollution and of the chemicals present in the environment. And this picture tells you how biosensor market is. Okay, percent revenues by end users from 2006 to 2016. I think with this, you people must have gained a basic knowledge on the types of biosensors, and these are some of the references. Thank you for your attention.